Welcome back to my channel Knowledge Chronicle.
Today we're diving into the intricate world of flowering plants, the backbone of life on Earth. Specifically, we'll explore the fascinating anatomy of dicotyledonous and monocotyledonous plants. So grab your notebook and let's get started. Before we get into the details, let's understand what is plant anatomy. It's the study of the internal structure of plants. Every part of a plant, roots, stems, and leaves has a specific structure tailored to its function. Today, we'll uncover these structures layer by layer. Ready? Let's begin with the dicotyledonous root. Take a look at this cross-section of a sunflower root, a classic example of a dicot root. Here's how it's structured. 1. Epidermis, the outermost protective layer. Some cells grow into root hairs. These tiny extensions absorb water and minerals from the soil. 2. Cortex, a multi-layered tissue made up of thin-walled parenchyma cells with intercellular spaces for gas exchange. The innermost layer of the cortex is called the endodermis. 3. Endodermis acts as a checkpoint. It's a single layer of barrel-shaped cells with waxy Casparian strips that control water flow into the vascular system. This ensures only essential nutrients enter the plant. 4. Pericycle, found just inside the endodermis. It's where lateral roots originate, and during secondary growth, this tissue develops into the vascular cambium. 5. Vascular bundles. The xylem and phloem form patches, usually 2 to 4 in number. The pith is either very small or absent in dicot roots. Key takeaway: dicot roots are designed for efficient nutrient transport and secondary growth. Amazing, right? Now, let's compare this with a monocot root. Think of plants like maize. The overall structure is similar to dicot roots, with epidermis, cortex, endodermis, and pericycle. The major difference? More xylem bundles. While dicots have 2 to 4 bundles, monocots have 6 or more. The pith is large and prominent, but here's a key point. Monocot roots don't undergo secondary growth. In simple terms, monocot roots are built for rapid water transport in short-lived plants like grasses. Moving on to the dicot stem. Think of sunflower or mustard plants. Let's break it down. First, we have the epidermis. It's the outermost layer, coated with a protective cuticle. Tiny hair-like structures called trichomes and stomata are often present. Next is the cortex, which is divided into 3 zones. The hypodermis, made up of parenchyma cells, provides mechanical strength. Then, there are the parenchyma layers, which allow storage and transport. Lastly, the endodermis, rich in starch grains, is also called the starch sheath. After the cortex, we find the pericycle. It's found as semi-lunar sclerenchymatous patches above the phloem. Now let's talk about the vascular bundles. They're arranged in a neat ring, which is a hallmark of dicot stems. Each bundle is open, allowing secondary growth, and endarch, meaning the protoxylem faces the center. Finally, we have the pith. It occupies the center and is filled with large parenchyma cells for storage. Now, let's talk about monocot stems, like those in grasses and sugarcane. First, we have the sclerenchymatous hypodermis, which provides extra strength and rigidity. Next, we see the vascular bundles, which are scattered throughout the ground tissue, unlike the ring arrangement in dicots. Each bundle is closed, meaning there's no secondary growth, and it's surrounded by a sclerenchymatous sheath. Additionally, water cavities are present for efficient transport. Dorsiventral leaves, like those of mango trees, show unique adaptations for photosynthesis. Let's start with the epidermis. The upper, or adaxial, and lower, or abaxial layers come with a protective cuticle. The lower epidermis usually has more stomata for gas exchange. Moving on to the mesophyll, this specialized tissue for photosynthesis is divided into 2 parts. First, we have the palisade parenchyma, which consists of vertically arranged, chloroplast-rich cells. Then, there's the spongy parenchyma, made up of loosely arranged cells with air spaces. Lastly, the vascular system veins contain vascular bundles surrounded by thick-walled bundle sheath cells. The size of these bundles corresponds to the vein size. Finally, let's look at monocot leaves, like those in grasses. Key differences include stomata on both sides of the leaf. Also, there are bulliform cells, which are large, empty cells on the upper epidermis that help the leaf curl during water stress. The mesophyll is not differentiated into palisade and spongy parenchyma. Veins and vascular bundles are nearly uniform in size, reflecting the parallel venation pattern. And that, my friends, is the intricate anatomy of flowering plants, a perfect blend of form and function. If you found this video informative, don't forget to hit that like button, share it with your friends, and subscribe to Knowledge Chronicle for more deep dives into the world of biology. Your support helps us create more valuable content. Until next time, stay curious.